Hey guys, Dr. McDonald. Let's do chapter 3. This is all about cells. For most of you guys who have had AMP1 in order to be in this class, this is going to be a review. I understand that some of you guys may not have had this recently. It may have been a while, so we will review it, but you should pick up on it fairly quickly. Okay, first thing, well, the cell. You can see various cells there, and I'm not going to go into any detail. It's more decorative. In a human body, we have about 100 trillion cells, and we actually have 10 times more bacterial cells than we do our cells. That's for every 100 pounds of your body weight, you have 1 pound of bacteria. 1 pound of bacteria takes a lot to make. And they're saying that 50 cells, that little eye right there, would contain 50 cells, so they're small. Fundamental unit of life is the cell, and it is the smallest unit that has all of the basic characteristics of life. And we remember the plasma membrane, cytoplasm, the genetic material, DNA, RNA, ribosomes for making proteins. And we've already talked about that there are two great categories of cells, prokaryotic and eukaryotic. So the microbes are going to be, a lot of them are going to be prokaryotes. We'll just go down the list. These guys are acellular. They're actually not alive. These guys are more in the category of infectious chemicals. Of all the cellular microbes, you have the eukaryotes, the algae, the protozoas, and the fungi. And then you have the prokaryotes, the bacteria, the cyanobacteria, and the archaea. What are the basic characteristics of life? All life must grow, must reproduce themselves, must be responsive. So it must be repelled by noxious stimuli and attracted by things like food and warmth or whatever it needs. Metabolism, it's biochemistry. It does chemical reactions. And then last but not least, it has a cellular structure of some sort. And they're asking that question. Well, that's the acellular guys we just saw. The cell theory, the classic cell theory, these guys came up with this. And you can see that they each kind of made a contribution, that plants were made up of cells, animals, and then Mr. Virchow, I think it was Dr. Virchow, found that all cells come from other cells. Robert Hooke was an Englishman who actually saw in cork, just regular cork, saw these structures like this, and he's the one who gave it the name cell. Reminded him of the cells of a monastery. Endosymbiotic theory, it's just a theory, nobody knows. But they're thinking that, okay, there's the eukaryote. We're not going to go to a lot of that. But what this basically says is that a prokaryote actually came into and lived inside the eukaryotic cell in a symbiotic relationship. And what this is meaning, and they're saying that this aerobic bacteria right here is the one who did it, and that these aerobic bacteria later on became the mitochondria that we have. There's also a similar situation with photosynthetic plants that have something similar. But there's some thinking that that's the origin of the mitochondria. What they're basing that on is the fact that the DNA of the mitochondria, if you recall, is separate from the DNA of the cell at large, is actually more of the type that you find in bacteria than you do in human cells. So there's some something to go with that, but it's just a theory at this point, and it's interesting, but that's about it. Prokaryotic cells, the bacteria and the archaea, we already said they don't have a nucleus. They have a nucleoid region where the genetic material of the DNA hangs out. They don't have a lot of those organelles, especially the ones that are surrounded by membranes. These guys are about 1 micrometer or less in diameter. So again, eukaryotic cells, prokaryotic cells. Most of you guys may have seen human cells under the microscope and A&P. When you look at the prokaryotes, you're going to be shocked at how tiny they are, even under the highest magnification. We already talked about these guys reproducing by binary fission, which is very similar in some ways to mitosis. I know they don't have nuclei. That's nucleoid regions, guys. But in any case, that's kind of what happens with binary fission. Cytoplasm is surrounded by the plasma membrane. Usually, most of them have a cell wall. Not all of them. There are exceptions. And then some of these guys, and I think I've already told you, maybe it was in the lab, that it may have a capsule, or it could have a slime layer. What's the difference? Capsule is a little more organized. Slime layer is fairly similar, except that it's not quite as organized or symmetrical. Prokaryotic cell structure, the fimbriae, these little hairs, plasma membrane, the cell wall, capsule if it's present. Pilus, these are like elongated fimbriae, and those guys are used for reproduction. And then the flagellum, which is used for motion. There's the nucleoid region right there, and plasmid is bacterial DNA is circular, by the way, although that doesn't look circular. If you took that circle and, well, it is circular. Then they have a smaller chromosome, which is called a plasmid, and that's kind of significant because that's where they carry a lot of the genetic information for drug resistance, for example. Cytoplasm, ribosomes, they have to make protein too. But I guess one of the things that maybe if you recall from Anatomy and Physiology 1 is just how simple these are by comparison to eukaryotic cells such as humans. Okay, morphologies. These are important. This is something that we're kind of touched on in the labs. So this spherical is called a coccus. The plural is cocci. This rod shape, a lot of times we use the word rod, but the term is bacillus and the plural is bacilli. The reason we kind of shy away from that is we've got actual group of bacteria whose genus name is bacillus. That wasn't a good idea. It's very confusing. But sometimes you'll hear it as bacillus. So if I talked about Bacillus cereus, you know I'm talking about a rod-shaped bacteria. It's a member of the genus Bacillus. But there are a lot of rods that aren't in the genus Bacillus, so that's the confusing part. Vibrio is a curved rod. This is like the disease-causing organism for cholera. Cocobacillus is somewhere that's between a round spherical cocci and a rod. So they give it a name between the two. This guy is the spirillum. He's more wavy like this, whereas the spirochete is more corkscrew shaped. That's the best way, I think, to keep these two guys apart. Now, bacteria, a lot of times, sometimes you will see them all by themselves, like that single coccus. You can have two of them, which they call a diplococcus, a tetrad, which is 4, streptococcus, the guys who give us strep throat. Strepto means chain. Staphylococcus is clustered up. It actually looks like a bunch of grapes. Bacillus, we've talked about. Streptobacillus is rods that form that same chain. You need to know all of these guys, so please make a special note to have this down pat. Okay, this again we've talked about in anatomy and physiology somewhat. Okay, so how do the cells maintain their shape? If it's isotonic, remember, in other words, if I have the same number of particles dissolved in the fluid outside the cell as I do inside, it's isotonic. Water is not going to move. We're not going to have any osmotic movement of water one way or the other. Hypertonic, if the solution outside has way more than the one inside, then the water will move out to try to dilute it. And that's going to get that crenation where the cell shrinks. Now for bacteria, this is a special case. Let me go finish going through this and I'll tell you. And then again, if you have it so that it is way more concentrated inside the bacteria, then water would tend to flow in. Now, we said all that, but remember, unlike our human cells, which will swell and burst or shrink, these guys are surrounded by a cell wall. In a hypertonic solution, they can lose their water and crenate. But in a hypotonic solution, the cell can only swell until it reaches the wall and then it's done. Think about it this way, guys. Everybody's seen a water balloon. You hook it up to a water faucet, you pour water, and if you keep pouring water, eventually the thing bursts. Okay? What if you took that same balloon and put it like in a bottle, some sort of bottle of soda or whatever? You pour it in there, the balloon is going to expand until it reaches that hard wall, and then it's going to stop. That's what happens with bacteria. Why am I going out of my way to tell you this? Because a lot of the drugs that we use to kill bacteria break that cell wall. And if we break that cell wall, then these guys go down to just like this. They will actually swell and go pop, and it kills the bacteria. Okay, continuing onward. Oh, well, they actually did all this on this side. That's actually really good. And you can see how this helps them. Plasmolysis. All right. Let's go to the next slide. Prokaryotic chromosomes. Again, bacteria generally only have one chromosome. They may have a smaller plasmid, a single long circular nucleoid regions. We already talked all about that. That's a good picture though. Prokaryotic ribosomes. This is one of the differences between prokaryotes and eukaryotes. The small subunit is 30S. The large subunit is 50S, and when it's together, it's a 70S. If you remember, in eukaryotes, it adds up to 80S, and these are also larger. Inclusions. These are things that are stored inside chemical droplets, uh, sulfur, magnetosomes, gas vacuoles. That's all an inclusion is. It's not an organelle. It's actually just a bubble that is storing something that is needed. Endospores, we talked about this. I told you it was a cross between a tank and a lifeboat. So this is what happens. Some, like the bacillus and the clostridia, can make these spores. These spores, when the conditions are not right for the bacteria, if they're going to die, what they will do is they will form one of these cells, spores in their body. They will duplicate their DNA and whatever else they can in there, and this guy is a tough nut. And then this cell will actually break apart and die and leave this spore. Those spores are hard to kill. They're not impossible, but they're very hard. And this is what makes these guys, especially the anthrax, so dangerous. Clostridium perfringens make spores. Uh, there are four. Well, we'll go through those. I don't want to go through all of them. But these spores are dangerous. This is one of the things you get food poisoning from, bacterial spores in the food. It doesn't matter if the bacteria themselves are dead. These spores, which are armored and hard to crack, when the conditions are right for the bacteria to live and grow, they will come out of there and they will start growing. So you can see this is a really easy way to get infection. These bacterial spores for the Clostridia species are all in the soil, and that's where these spores from gangrene come from. Now, these guys, specifically Clostridia, are anaerobic. They can't live in an oxygen atmosphere, but this is what causes gangrene in our patients with diabetes. So sporulation is the way that they form a spore. It's not really reproduction because it's not binary fission. It's actually like a lifeboat, but it's an armored lifeboat that is very tough. These guys can survive for years. They are resistant to heat, to cold, to drying, to alcohol, to a lot of chemicals. When we get, there's a Clostridia species called difficile. It causes really bad diarrhea and can oftentimes kill people. When we have a patient like that, we do not use that alcohol foam that we use everywhere else because the alcohol foam will not penetrate. It will not kill these guys, so you actually have to use soap and water, not to kill them, but to wash them off your hands. Usually one spore per bacteria, and we talked about in the lab about the endospore stains. Very important identification. Here's the process of sporulation. Replication, make a membrane, and then going through the process. And then there again is the picture. The pink is the vegetative cells and the green is the endospore. And I don't see anything else. Malachite green is the stain we use. Okay. Plasma membrane of a bacteria, not a lot different than it is from any other cell. A phospholipid bilayer. You've got proteins, integral and peripheral proteins. You've got these glycoproteins and glycolipids that are on the outside signaling. So you've got the cytoskeleton. So in a lot of ways, it's very, very similar to the ones that you studied in anatomy and physiology. And then there's the enzymes on the outside of it. Okay, not a whole lot more to say on that, to be honest. Bacterial cell walls. This is where we come to a difference. This is what the cell wall of there's a plasma membrane right there. That's the cell wall of a gram positive. And there's a membrane. It's got a phospholipids on the bottom, and uh, it's got a different lipid on the top. And then there's a very thin cell wall, and then there's the regular plasma membrane. That's one of the differences. So these guys, peptidoglycan, that's what you need to know. These guys are made of the NAM and the NAG. The NAG, N-acetylglucosamine, or the blue, the N-acetylmuramic acid, the NAM, or the yellow. And then you've got these groups of amino acids that connect them together. You got the tetrapeptide that connects them vertically, and the pentapeptide that connects them horizontally. A lot of our antibiotics work to mess this up right here. Different ones will strike different ones, and we'll talk more about those when we get there. And there is the one for the gram negative. They've got the NAG and the NAM, but these guys have direct links. They don't have these pentapeptides. And mycoplasma don't have cell walls. They're pleomorphic, which means they don't have a certain shape. We'll talk more about those. So remember peptidoglycan, remember the NAG and the NAM. Going to be important stuff. Here's a good. So there's the plasma membrane. There's the big thick wall in a gram positive. Plasma membrane, there's a space, and then here's the cell wall, and then here's another membrane. The bottom part of this is going to be a phospholipid. The top part is something called lipid A, or Lippo Polysaccharide. These guys make your immune system go absolutely nuts. This is what causes septic shock, because when this is present in your bloodstream, you can have your entire immune system is going to go into an over response that's going to dilate all your blood vessels and cause your blood pressure to drop to virtually nothing. So you can see the problem. So these guys also have this teichoic acid that comes up from the plasma membrane, helping attach it. So what we do is we try to break these. Now this is a little bit easier. This one, you have to go through a plasma or a membrane, which is going to reject any kind of charge, and then you get to a wall, and then you have another one. And this is why, in general, gram negatives are a little bit more difficult for us to kill than the gram positives. These are the most common disease causing. These are the worst to get. Okay. Mycolic acid. These are the acid fast. When you think of that, you can think of tuberculosis. Mycobacterium tuberculi. Anyway, these guys, they have the plasma membrane, they have a peptidoglycan cell wall with the teichoic acid. And as a matter of fact, they look just like gram positives, but then they have this mycolic acid. This mycolic acid is a lot like wax. This is why it's so hard to kill tuberculosis, those waxy cells. Now, on the other hand, it makes these guys grow very slowly because they're very complicated, but that's one benefit we have, but they're very difficult to kill, and they have other things to protect themselves too. Okay, talking about our lipopolysaccharide, the lipid A is in there, and then there's an O side chain. This is extremely immunogenic. It's going to cause your immune system to go crazy, and that's a toxic shock. Septic shock is actually the term. Glycocalyx, we talked about that in anatomy and physiology. Well, here these guys are going to be the slime layers in the capsules. And we saw in the lab where we have this space, the cells are stained and there's a stain in the space between the cells, but then there is a cleared area between the cell and that can only be a capsule. This is going to protect the bacteria and make it much harder for us to kill them. Encapsulated bacteria it's more difficult for our antibiotics to get to them to destroy them. Slimy, gelatinous, secreted outside. Slime layer is loosely connected, disorganized, and then the capsule. I've already talked about that. Capsule stain of Pseudomonas aeruginosa is a bad guy. He's gram-negative, and he's got a capsule. That's pretty bad. He also gets involved in a lot of stuff. You'll see him a lot with burn patients or patients that have infections that came from water. Those Pseudomonas has been known to live in some of the disinfecting liquids we use in a hospital. Pili and Fimbriae. There's the pili. The pili, well, let's talk about the Fimbriae. They're numerous. They're shorter than, of course, the pili or the flagella. Hair-like, gram-negatives. These guys actually, those Fimbriae actually help the bacteria to attach to surfaces. And being able to keep from being washed out, say in the GI system or anywhere else, is a big advantage. It's a virulence factor that makes them more difficult. And then you have the pili. These guys are, well, the anchoring also helps with biofilms. We'll talk about that in a little bit. The sex pilus is the one that allows the bacteria to transfer genes from that small chromosome to another bacteria, even bacteria of a different species. And by doing that, that's how antibiotic resistance spreads amongst bacteria is plural, excuse me. Flagella, we talked about this as being the mechanism of motility, how these guys do it. Seeing a gram-positive and a gram-negative. Whoops, I lost my thing. Oh well, I won't worry about it then. Any case, these things are anchored on that inner membrane and it goes through the wall. And these guys are going to spin around like a boat propeller. And by doing that, it's going to move them just like a propeller moves a boat. And you see that's a little bit different in the gram-negative, but for our purposes, that's good. How the flagella, monotrichous, at the end, amphitrichous, you got them at both ends, and those are oftentimes called polar flagella. Iapotrichous, you've got a bunch, but it's only at one end, and paratrichous is where you have these flagella all over the bodies, and they come together in groups. You can see all that right there as well. Okay, guys, let's see. Oh, motility. So these guys are going to, when their flagella are rotating clockwise like this, these guys oftentimes are going to tumble, and by tumbling I mean exactly that. They'll do this, and then they will rotate these guys counterclockwise, and they'll all be together like that. And when they do that, they have a run. So bacteria will oftentimes tumble and then make a beeline, and then tumble and make a beeline. and they're moving in response to stimuli, taxis. They're moving toward food or away from things that are noxious to them or dangerous. Here's a good example right here. It's making a run counterclockwise. You see, it's going to separate these guys, and then they start rotating clockwise, and it's going to tumble, and eventually this guy goes this direction. I know it doesn't look like it's the most effective and efficient way, but it's their way, and apparently it works because they are highly successful. Without the chemical gradient, which is something either noxious or something like food, these guys will cycle between the runs and the tumbles and have no real directional movement. It's almost like a random checking out of different spaces to see if there's anything there that they either want or something they need to avoid. Chemotaxis is the moving toward a chemical concentration, with a higher concentration being where they're moving. Eukaryotic cell, this ought to be absolutely familiar, but algae, protozoa, fungi, animals, plants. Plants have walls. Algae, fungi have walls. Protozoa sometimes have walls. Animal cells do not have walls, and again, sometimes protozoa don't, some of them don't. Anyway, 10 to 100 times larger than the bacteria, 10 to 100 microns. More complex membrane-bound organelles. I mean, that's about the size of a bacteria right there to give you an idea of the scale. I'm not going to read through all this again. This is review. Look at it, and it should come back to you pretty easily. Eukaryotes, we've got a nucleus. Well, I can't do it there. Prokaryotes don't. Eukaryotes, we've got plant and animals and other types. The animals don't have a cell wall, and plants have one, but it's different, of course, from the bacteria. Eukaryotics have those membrane-bound organelles. Prokaryotics have no membranes other than the membrane that surrounds them. Okay, guys, that's everything for the first week, and I will be getting chapters 4, 5, and 6 up for you. Again, bring your questions to the meetings, scan through the textbook, and look in the textbook after you've done this if you see any questions, and then you can ask me too, of course. All right, guys, that's it for now.